Ryan Trometta, University of Rochester, USA.
Every time a doctor performs a surgery, a potential for complication multiplies. Surgeries performed on bone risk the common occurrence of bacterial bone infections, known as osteomyelitis. This prevents the bone from healing, can lead to sepsis, it can lead to limb amputation. The current treatment for osteomyelitis is a two-stage surgery. First, doctors remove the infected bone in a process known as debridement. Then they insert cement beads filled with antibiotics to eliminate any remaining bacteria. This local drug delivery enables a high concentration of drug to reach the infection site, which is not possible when the drugs are either taken orally or systemically due to toxic side effects to organs. You can think of this as the same as when you fall down, let's say, bruise your elbow. You don't go home and take a full-body ice bath because you would freeze. Instead, you ice your elbow and only your elbow because that is the precise location that needs treatment. This is the same concept. These beads remain within the patient for a few weeks or even months and then are removed in a second surgery, and the doctors insert a bone graft to initiate and enable bone healing. But this current clinical standard has serious flaws. One, it requires 2 surgeries, which increases patient costs, care, and time. And even after all this, the bacteria can still survive, resulting in a relapse of the infection. So why do these bacteria still survive? Well, it's actually because they exist in 2 different states, a planktonic state and within a biofilm. The planktonic state is susceptible to antibiotics, but the biofilm acts as a cocoon, shielding the bacteria within from the antibiotics. However, in research, we have developed biofilm dispersal agents which could break down the wall of the biofilm and allow the antibiotics to go in and eliminate the bacteria. But now the question is, how do we incorporate both antibiotics and biofilm dispersal agents locally and at high concentrations in the most efficient manner? Well, in our lab, we have a solution, and that solution is 3D printing. With 3D printing, we can print precise patient geometries from clinical scans. As shown here in the video, in which we're printing a model of a patient's spine who has severe scoliosis. But we just can't only print models, we can also print biocompatible bone grafts, which could be inserted into a patient. We can also print directly antibiotics and biofilm dispersal agents directly into these bone grafts, such as the one that I have here in my hand. So when an infection does occur, we can remove the infected bone, then insert our bone graft in one single surgery. The biofilm dispersal agents will break down the barrier of the biofilm so the antibiotics can eliminate all the bacteria. This will prevent a future relapse of the infection. And once the infection is totally gone, the bone can completely heal. Thus, with this 3D printed bone graft with both antibiotics and biofilm dispersal agents, we will not only break down the wall of the biofilm, but break down the wall of osteomyelitis.